Welcome to Prescribed by Dr. Nathan Goodyear, where integrative oncology meets holistic healing. This podcast is your home for the latest in innovation, exploration, and elevation in all areas of integrative oncology, medicine, health, wellness, and beyond. Join me and a host of other innovators as we go from practicing to prescribing. Your daily dose of wellness begins now. Intratumoral vitamin C. What if that was cancer's kryptonite? Go back to the movies of Superman. What if Superman's kryptonite was cancer's vitamin C? Welcome to the Prescribed by Dr. Nathan Goodyear podcast. You know I love to talk about all things vitamin C because vitamin C, it is the flagship of integrative medicine, and really it's the flagship of integrative oncology. And we are at a precipice. We are a precipice of change where vitamin C, the research is really becoming so strong, really overwhelming to its benefit as a standalone therapy, as a precision therapy, but as a therapy in conjunction with conventional oncology therapeutics, such as surgery, chemo, radiation, immunotherapeutics, immune checkpoint inhibitors, targeted therapies. And we're at that reflection point in the research. The problem is doctors don't know it because doctors don't read. So that's what this podcast is for. I'm super excited to bring this new content of where I think we're going next with vitamin C. So when we look at vitamin C, great quote from an article, check it out in the references below. The quote, "The biological functions of vitamin C can be attributed to its biochemical property as an electron donor." See, when we think about vitamin C, most people think about it as a vitamin, and that word vitamin is two words connected together, vitamin. And these are way back in the 19th century where they thought that life was essentially made up of these amines. So these amines were vital to life. And that's where the word vitamin came from. And that's what vita in Latin means. It means vital. And so here they are vital for life. So vitamin C, yeah, we think about it, it is vital for life because vitamin C deficiency is going to lead to scurvy and it can take your life. But when we actually look at the foundational aspect of what vitamin C does is its ability to donate electrons. See, we think about vitamin C, pharmacological vitamin C. We don't think about vitamin C as a pro-drug, that's hydrogen peroxide. We don't see it as a drug, but it really is. We don't see it as an electron donor because we don't think about the reduction oxidation process much. We don't think about dehydroascorbate, which is the oxidized form. We don't think about the reduced form, which is ascorbic acid. We just think about vitamin C. And so what we have here is the complexity of vitamin C that you're going to get from this podcast, from this video if you check us out on YouTube, but your doctor probably doesn't. And so sit back because you're going to get to know more than your doctor. So another famous quote from Linus Pauling. I love Linus Pauling. One of the two individuals that won the Nobel Prize of Medicine Physiology solo, individual. No others that were part of his team. It was him. Now, his original research, 1976, 1978, with vitamin C in terminal cancer patients was with Ewan Cameron. And Ewan Cameron was a he was a Scottish, big slip there. He was a Scottish surgeon. So it was Linus Pauling and Ewan Cameron. And what they did together was they looked at these terminal cancer patients, gave them 10 grams IV vitamin C. And they did this in 1976, and then they reproduced it in 1978 and actually found greater value. But famous quote here from Linus Pauling said, "Although physicians, as a part of their training, are taught that the dosage of a drug that is prescribed for the patient must be very carefully determined and controlled, they seem to have difficulty in remembering that the same principle applies to vitamins." And so when we look at a lot of the natural, holistic, and integrative therapies, one of the things that concerns me is that the people designing these studies don't understand these pharmacokinetic and pharmacodynamic principles of these natural, holistic, and integrative therapies. They do in the conventional world because that's their lane. But what they do is then they take the research and they apply it to a lane that they have no business being in. And so you're seeing this in the mistletoe. And so we see this error be repeated time and time again. Thankfully, we've overcome the error with vitamin C. And I think we have now those that are pushing forward to make sure that same error doesn't occur in things like mistletoe. Now, we have to understand that the key to vitamin C is delivery. Delivering oral vitamin C is not the same as delivering IV vitamin C, which is not the same as delivering intratumoral vitamin C. Now, with Linus Pauling, New, and Cameron in 1976, 1978, they gave IV 10 grams vitamin C. Then there were a group of physicians at the Mayo Clinic that decided to reproduce that and only give oral vitamin C. And of course, they published that they found no statistical benefit in the cancer patients. And so then mainstream or legacy media took that up, and there was no counter, new digital media to counter the narrative that they set. And then so for the next basically 20, 30 years, vitamin C was thought to be non-effective because a group of doctors at the Mayo Clinic didn't understand the difference between oral and IV pharmacokinetics, what's called LABI, liberation, absorption, distribution, metabolism, and excretion. It's going to be different between the oral and IV route. They missed it. So you can see why I'm concerned about the research. That dominated for 20, 30 years. We can't let that happen again. So when we look at oral vitamin C is really limited by what's called two-compartment pharmacokinetics. Now, what's that mean? It's a pharmacological principle that as it relates to oral vitamin C, we're probably not going to achieve more than 70 to 80 micromolar of vitamin C in the plasma. And it's this two-compartment pharmacokinetics that keeps that there. That's that kind of set point. And the way it does that is when you're taking vitamin C in by mouth, as it travels down through the gastrointestinal tract, and your plasma vitamin C is less than 70 micromolar, then it's going to upregulate the GI absorption of that vitamin C. Absorption, that's the second component of that acronym LABMI. And then what it's going to do is it's going to also reduce the renal excretion of the vitamin C, so it's going to increase the plasma ascorbic acid level. But if that plasma ascorbic acid level is, say, above 70 micromolar or very close to it, you're going to see a decrease in the GI absorption and an increase in the renal excretion to maintain that very steady course. Now, this is why oral vitamin C is never going to achieve a therapeutic direct cytotoxic effect against cancer. It never will if given orally because of that two-compartment pharmacokinetics. Now, people will say, yes, you can, but basic pharmacokinetics of vitamin C says, no, you cannot. And two-compartment pharmacokinetics puts the exclamation point on that, no, you cannot. And so there's actually been some research done that actually looked at the different plasma ascorbic acid between the oral and IV route. And so, and you look in the show notes, you'll be able to see that there. Let's see, do I have the reference right here in front of me? I don't, but it will be in the show notes, so definitely check that out. So by looking at the oral vitamin C, there was this study, direct comparison. They looked at oral doses of 0.03 grams per day, 0.1 grams per day, 2 grams per day, 0.4 grams per day. And they looked at maximum tolerated dose of 18 grams per day, which was at 3 grams every 4 hours. Now, what they found is that the plasma concentration in micromolar at 0.4 grams at an individual dose only achieved 70 micromolar plus or minus 4.4. That's it. So there's that 70 to 80 micromolar. Now, when you look at the IV dose, okay, and if we look at this from really two perspectives, one is a gram per kilogram dose and a total dose. So if we looked at 0.1 gram per kilogram of IV vitamin C, it achieved 2.4 millimolar, not micromolar, but millimolar. So significantly higher plasma concentration. At 1.5 grams per kilogram, which many, including myself, believe this to be the high dose value, it achieves 26.2 plus or minus 4.9, or right around 30 millimolar. Now, why is this important? Research shows if you want a direct cytotoxic effect against cancer, you need to achieve a plasma millimolar range of 20 to 40 millimolar. But the foundation, the bare basement level is 1 millimolar versus 70 to 80 micromolar with the oral dose. It's not achievable by the oral route. Now, you can modify the gut microbiome. You can create epigenetic issues or epigenetic benefits. So there is some benefit there, but I would tell you that there's some research that says be very careful with that, because in cancer, that suboptimal vitamin C dosing there on a massive scale could actually favor the cancer. But then when we look at the vitamin C dosing at 60 grams per week, the millimolar dose is 21 plus or minus 5. So significant therapeutic value when it's IV, but not oral. And it's because of that 2-compartment pharmacokinetics. Another point to recognize, so that's talking about oral versus IV. Okay, so if we're talking about direct cytotoxic, cancer-killing effects, it's vitamin C. Now, vitamin C can have other effects, immunomodulatory effects, anti-angiogenic. You can use it with other, conventional therapeutics. But we have to recognize that when we're using vitamin C from the IV route, we can't just treat everybody alike. You recognize this with a 5'2", 120-pound patient sitting next to a 6'6", 366-pound patient. Intuitively, people go, why are we both receiving 50 grams? That person is as big as my thigh. That is the 5'2" talking, the 6'6", 366-pound talking about 5'2". Intuitively, they recognize that their size dictates a different response. And you see this in conventional pharmaceuticals. You see this. Bigger people are typically going to need a higher dose. And you see this in IV dosing of pharmaceuticals as well. But when we look at vitamin C in the IV route, we have to recognize that beyond the weight, the size of the individual, there's things like tumor burden. How much burden is present? Is it just one tumor that's 2 to 3 centimeters, or are there multiple tumors all over the body? What's the vitamin C deficit of the individual? When you look at cancer and vitamin C, it's really kind of a metabolic scurvy situation. Many of the cancer patients that I see, they're massively deficient in vitamin C. So you give them any vitamin C, the body is going to use what it needs to. And so it's going to require you to dose higher if there's a massive vitamin C deficiency. Understand cancer would also be considered a vitamin D deficient state. So it would also be a rickets, metabolic rickets, if you will. The metabolic demand delivery. So the metabolic requirements of the body, guess what? We need lots of vitamin C because we as humans don't make it. We lack the enzyme glutolactose oxidase to make it. The redox balance, the reduction oxidation balance. Remember, vitamin C is an electron donor. And whatever that redox balance is, the vitamin C may or may not need to be dosed higher. And then what's the level of the inflammation? So if this is a cancer situation where there's massive inflammation ongoing, it's out of control, or patients receiving a pro-oxidative therapy like chemo, radiation, or even a natural therapy like low-dose metronomic chemo, and yes, vitamin C can be pro-oxidative. You don't want to overwhelm the oxidative stress situation because that will aggravate the inflammation. So in that presence, if there's a lot of inflammation, the vitamin C dosing may need to be elevated to counter the inflammation response that vitamin C would counter. But also understand the therapeutics involved could also require a different dosing of vitamin C. And that could be up or that could be down. Because if there's overwhelming oxidative stress that has been detected in the body, and you can see that in, say, high sensitivity C-reactive protein sedimentation rate, you might want to tone back the pro-oxidative vitamin C dosing. But if you don't see that and the patient has a coexisting oxidative therapy like radiation, you could add pro-oxidative vitamin C with that to match the tumor burden, the vitamin C deficit, the weight, the metabolic demand, and the redox and inflammation. So when we look at the oral and the 2-compartment pharmacokinetics, we have to understand in there it's primarily serving as an antioxidant. It's improving the gut microbiome balance, particularly with bifidobacteria, and it's an epigenetic modifier. But when we look at the IV routes, we're obviously getting increased systemic delivery, and we're getting pro-oxidative effect, immunomodulatory effects, and we're getting these pharmacokinetic benefits by bypassing the gut and going right into the IV. And by doing so, we get into the plasma where the ascorbic acid is. Now recognize that what needs to happen in the plasma when we deliver vitamin C is it must reach the site of action. It must reach the tumor, but it doesn't just go from the blood to the tumor. It goes from the blood into the extracellular space, from the extracellular space, then into the tumor microenvironment, and then it reaches into the tumor and it saturates the tumor, and it has to happen this wherever the tumors are. So we have to recognize that what happens is the plasma ascorbic acid that we give IV, it is actually converted to ascorbate free radical and then eventually converted to hydrogen peroxide. And research, actually a beautiful study published in 2007 entitled Ascorbate in Pharmacological Concentrations Selectively Generates Ascorbate Radical and Hydrogen Peroxide in the Extracellular Fluid in Vivo. In vivo. Not human clinical trials, but in vivo, so animal models here. And what they found is that the preferential formation of the ascorbate radical and hydrogen peroxide in the extracellular space did occur because it didn't happen in the blood because of the red blood cells and their detoxification enzymes not allowing that to happen. And so what was then happening is in the extracellular space, this gradient was allowing the production of these oxidative free radicals that then could generate eventually the hydrogen peroxide that then could generate the hydroxyl free radicals and the superoxide anions that could then lead to the pro-oxidative state in the tumor microenvironment and in the tumor, hopefully saturating all aspects of the tumor, the normoxic, the normal oxygen, and then the hypoxic, which is a low oxygen state. But this wasn't happening in the blood, and it was because in the blood, the red blood cell catalase and glutathione peroxidase was actually breaking down this production of the prodrug, which is hydrogen peroxide. But when we give it in proper dosage and account for those variables, tumor burden, weight, redox, inflammation, and we then dose appropriately, monitoring the plasma ascorbic acid levels, not the capillary glucose, but monitoring the plasma ascorbic acid levels, we can then reach the extracellular space, reach the tumor microenvironment, and reach the tumor. Now, we have no ability to test clinically in those spaces of extracellular space, tumor microenvironment, or tumor, but we do in the plasma, and we can extrapolate that out. But let's talk about where I think the future is going with vitamin C, and that's intratumoral. See, intratumoral vitamin C is tumoricidal, kills cancer cells. It's an immunomodulatory agent, and it's synergistic with chemotherapy. So it's tumoricidal, it's an immune modulating agent, and it's synergistic with chemotherapy. So I want to go through some background on this, just a few studies, and I want to talk about a recent study that really, I think, created some exciting future for intratumoral vitamin C. The first article is from 2012, and we're going to go back just a little bit, but effect of high-dose intravenous ascorbic acid on the growth of marine sarcoma in vivo. Now, the purpose here was to investigate the effects of high-dose ascorbic acid. Vitamin C here was done in both IV and intratumorally, okay, because they wanted to study the effects. And they were looking at mice that were implanted with sarcoma cancer cells. And these were to create tumor models within these mice. That's what a marine model is. It's taking advantage of the similarities between the mice and humans and using these mice as a means for analysis without, doing it in humans. And so what they did is they monitored tumor growth over the time to assess the impact of the different delivery techniques of the ascorbic acid. And here were the key findings here. They found high concentrations of ascorbic acid induced cytotoxic effects on the tumor cells. Translated, slowed the tumor growth. The intratumoral administration resulted in higher localized ascorbic acid concentrations compared to systemic delivery. That makes sense, right? If you go right to the tumor, you're going to be able to deliver more ascorbic acid, or more of anything for that matter, unless you just automatically reduce the dose, which, by the way, you can, because you're going right to the tumor. And you're completely bypassing the limitations of that plasma to extracellular space, to tumor microenvironment, to totality of tumor, and then tumor burden. You're bypassing all of that. It's like chemotherapy. If you go right into the tumor, it bypasses the off-target toxicity of the systemic chemotherapy. I mean, it makes sense. But here, actually, in study of mice, they actually showed that the intratumoral administration achieved higher local vitamin C concentrations compared to the systemic delivery. Yet, what you now heard there and now know from a 2012 study? Good luck finding a doctor that knows that. Good luck. And that's my problem, because we doctors must teach doctors. And it's a science problem, because science is not about propagating a narrative. It's about propagating the science, the truth. The pro-oxidative mechanism of the vitamin C was generated by the hydrogen peroxide. And this intratumorally or systemically was still proven to be the case, the primary effect here of the cancer-killing effect. So the conclusion was this: high dose ascorbic acid, particularly through the intratumoral delivery technique, demonstrated significant anti-tumor activity in vivo, suggesting its potential as a localized cancer therapy. 2012. Now, the limitation was this was an animal model, and it's not a human model or a human study for cancer treatment. So we have to recognize that animal studies don't always translate to humans, but we have plenty of human studies of the systemic benefits of vitamin C that, you don't want to say take it to the bank, but take it to the bank. Then the next study here is, we'll go back just a little bit more, 2008. Pharmacological doses of ascorbate act as a pro-oxidant and decrease growth of aggressive tumor xenografts in mice. Now, the objective here was to examine the effects of the pharmacological doses, I've used that word before, of vitamin C on the growth of aggressive tumor xenografts. Now, tumor xenografts are a transplant of an organ, tissue, or cells to an individual of another species. And this was established in immune deficient mice, and they implanted aggressive cancer cell lines. So they took human cancer cells and implanted them in immune deficient mice. The higher doses of ascorbate, that's ascorbic acid, vitamin C, were administered then IV and intratumoral. So again, looking at both. The study measured tumor growth, survival rates, and biochemical markers of oxidative stress. So this study was looking at both in vitro and in vivo. So here are the key findings of this second study. High dose ascorbate selectively decreased tumor growth without affecting normal tissue. Boom. See, I've talked about this a lot. Natural therapies have the capacity to target abnormal cells in abnormal areas and protect and preserve healthy areas. The therapeutic effect was mediated by ascorbate-induced hydrogen peroxide formation, which caused oxidative stress in the tumor microenvironment. Normal cells were left unaffected, and it was due to their robust antioxidant defenses, just like the IV in the blood. It had the catalase, the glutathione peroxidase. Same thing in healthy cells. So giving vitamin C, it can be pro-oxidative and antioxidative in the same body. Intratumoral delivery achieved localized pro-oxidative effects with enhanced tumor cytotoxicity. So 2 studies looking at both. Third study, the potential use of ascorbate in the treatment of cancer, published in Anti-Cancer Research 2009. The objective here was it was a review to look at the potential of ascorbic acid as a therapeutic agent in cancer treatment. And they looked at different mechanisms of action, but at the pharmacological dose, ascorbate generates hydrogen peroxide in the tumor microenvironment. Already referenced that. And it induced oxidative stress and cytotoxicity in cancer cells that have deficient detoxification enzymes, particularly catalase and glutathione peroxidase, and even superoxide dismutase. Both systemic, that's IV, and localized intratumoral delivery methods were discussed in this review article. And they discussed that intratumoral administration probably was the best delivery technique because of, again, the obvious, the direct, high localized concentrations delivered right there. Pretty easy, right? Pretty easy. So their potential benefits they recommended was the synergistic effect of chemotherapy and radiation, meaning the benefit of pharmacological vitamin C, whether at IV or intratumoral, with chemo and with radiation. Talk to your medical oncologist. Talk to your radiation oncologist. They're going to tell you not to because it's an antioxidant. And here's the research that's saying, no. All to the contraire. Read the research. It's minimally toxic to normal tissues, even at high doses. Let me repeat that. Minimally toxic to normal tissues, even at high doses. Why? Because the high doses have the capacity to detoxify it. They have the capacity to handle the electron donation, redox buffer. The cancer cells don't. And it has anti-angiogenic properties, reducing tumor vascularization. So all of that background, that history of those studies talking about intratumoral, to bring you to a recent article entitled In Vitro and In Vivo Assessment of High-Dose Vitamin C Against Marine Tumors. Now, this was the animal model, and they looked at female mice aged 8 to 10 weeks, and they used two types of marine cancer cell lines. So they employed breast cancer cells and colon cancer cells, particularly 4T1 breast cancer cells and C226 colon cancer cells. And remember, a marine model is cells that are not from mice injected into mice. And that's exactly what here. They took human cancer cell lines and injected them into the mice. So it's one species to another to follow a biological phenomenon. Then what this did is induce tumor induction. And what we saw is the subcutaneous inoculation of these tumor cells into the flank of these mice grew tumors. And they followed these tumors until they reached 90 cubic millimeters in volume, in milliliters, okay? And this was typically about 8 days after the inoculation. And then they divided these mice up into three groups. The control group, which received no vitamin C, then the low dose group, which received 100 micrograms per mil of intratumoral vitamin C, and then the third group, which the high dose, which delivered 200 micrograms, which delivered and then received the 200 micrograms per mil of intratumoral vitamin C. So again, we had two parts to this study. The first start is you implant the tumor. You let the tumor grow. The second part is you say, okay, let's divide it up to the treatment group that receives no vitamin C, just placebo. Second is low dose intratumoral vitamin C. Third is high dose intratumoral vitamin C. Again, low dose 100 micrograms per mil, high dose 200 micrograms per mil, but understand they dosed it all the way up to 1,000 micrograms per mil. And these intratumoral injections were right into the tumors. This was not IV. So here's what they found. They found that both the low dose and the high dose vitamin C treatment groups resulted in significant delay in tumor growth compared to the control group. The high dose group exhibited a more pronounced tumor growth inhibition. So the control group, where they injected nothing but probably saline, no change in tumor growth, right? The low dose, actually, you had a reduction in tumor proliferation. The higher dose, you had a dose-dependent reduction in tumor growth. And no significant toxicity or adverse effects were observed in the mice receiving the intratumoral vitamin C, up to 1,000 micrograms per mil. The treatment was well tolerated and had a very high safety profile. See, that theme has been reproduce. Yet here we are talking about vitamin C again. The in vitro findings resulted in inhibition of tumor cell proliferation. That's growth. Now, this suppressive effect was seen within 24 hours, meaning from intratumoral vitamin C injection, we saw these cells' growth inhibition start at 24 hours. That's quick. And this persisted over time. So imagine you go higher dose intratumorally. You're going to see this effect occur early, and you're going to see it continue, probably dependent time-wise on the dose given. It induced apoptosis. And so the vitamin C, we know, can trigger programmed cell death here, apoptosis. And it did so in a dose-dependent manner, meaning the higher the dose, the more apoptosis it triggered, programmed cell death in these cancer cells. They showed that the increased early and late apoptotic cell fractures were associated with a higher vitamin C concentration. So the earlier the programmed cell death that occurred, it was associated with the higher vitamin C concentrations injected, again now, intratumorally. Other findings, the intratumoral vitamin C was synergistic with chemotherapy. Real quick. Synergistic is not additive. Additive is 1 + 1 = 2. So that would mean intratumoral vitamin C plus chemo equals 3. No. Synergistic is 1 + 1 = 10. It's an exponential benefit. Combining vitamin C with chemotherapy, that's intratumoral vitamin C with chemotherapy here, particularly the drug cisplatin, enhanced the anti-tumor effects compared to either treatment alone. It also had immunomodulatory effect. We talk about immunostimulating, immunosuppressive, but here it's modulating the immune system, which means it can have an immunostimulating effect here and an immunosuppressing effect here. And you go, wait a second, I've never heard of that. What about something called a selective estrogen receptor modulator, such as tamoxifen in cancer? It can have an immunostimulating effect with estrogen receptors here and an immunoblocking or suppressing effect with estrogen receptors somewhere else. So in the immunomodulatory effect of the intratumoral vitamin C, it found that it modulated the infiltration of T cells into the tumor microenvironment. It increased the infiltration of T lymphocytes. Now, what's important here is it required a competent immune system. So if you have an immune deficient mouse model, or you have an immune deficient individual, if we extrapolate it out, that we may not see as much of a immune infiltration of the T lymphocytes into the tumor microenvironment. But here's the other thing. It augmented it augmented the immune checkpoint inhibiting therapies. So the intratumoral vitamin C augmented conventional immunotherapeutics, particularly the immune checkpoint inhibitors such as Keytruda, Opdivo, Yervoy, etc. And you can find that reference from a 2020 study entitled High Dose Vitamin C Enhancers Cancer Immunotherapy. Now, what's also interesting here is they proved the positive, the intratumoral vitamin C. Then they proved some of the negative, which is they added N-acetylcysteine, and it countered the pro-oxidative effect of the intratumoral vitamin C in the tumor. Now, it lost this at the really high levels, at the high dosing, that is the 1,000 micrograms per mil of intratumoral vitamin C. But the N-acetylcysteine counteracted the pro-oxidative effects of the vitamin C intratumorally. I've talked about, N-acetylcysteine. It's not a great therapy to use alongside IV vitamin C when you're using it pro-oxidatively to try to directly cytotoxically kill cancer cells. The same also applies here in this mouse model, saying it also applies to intratumoral vitamin C as well. So different doses of vitamin C, different delivery methods of vitamin C. If we want to target cancer, oral route, no, it's not a good route. IV route, great route. Now, it does have some issues we have to bypass. All those variables of inflammation, of redox balance, oxidative stress, tumor burden, body weight, all of those things limit the variability. Tumor burden is probably one of the biggest one of the IV route. And that's why, the systemic to tumor route of delivery of therapies in cancer, I think, is flawed. It's what we've known. And, a lot of times what you see in medicine is what you see in so many aspects of life. You do what you know. It doesn't mean it's right. It's what you were taught. Maybe those who were taught, maybe it was wrong. They just didn't know it. It's what they were taught. But here, the intratumoral vitamin C in this 2016 study suggests we can kill cancer cells in a dose-dependent manner. We can augment chemotherapy. We can augment immune checkpoint inhibiting therapy. Now, that was from another study, but it's a standalone therapy, but it's a therapy that's going to work alongside other therapeutics by going intratumoral, starting at a low dose of 100 micrograms per mil and go up to 1,000 micrograms per mil. But here's the best part, is it'll augment other cancer killing therapies. It'll kill cancer cells. It'll modulate the immune system to do it. It'll augment immunotherapies, but it protects and preserves healthy cells. Win-win. Win-win. It's a great therapy. So here's the conclusion. The study suggests that high-dose vitamin C, when delivered directly into the tumor, it serves as a new therapeutic approach for advanced tumors for the ability to inhibit tumor cell proliferation directly, induce apoptosis directly in a dose-dependent manner, and to enhance the effects of chemotherapy. I would add immunotherapy as well. Direct intratumoral delivery of high-dose vitamin C effectively delays tumor growth in this marine mouse model. Now, that is a limitation. It is a mouse model. It's not a human model. Now, this was a breast and colon cancer. But given its low toxicity and accessibility, and I would say the reproducibility of the systemic IV vitamin C that correlates with the intratumoral here in this mouse model, This approach holds great potential as a novel therapeutic strategy for patients with advanced tumors. But I always love how studies do this. Further research is warranted. When does further research become enough? That's kind of the CYA. When you're really proposing a new idea, a new concept, you go, whew, boy, things might get really hot with that bullseye on my you-know-what. So let's throw in a CYA comment, and that is further research is warranted. That's the CYA comment. So the future of vitamin C? I'm a big fan of IV vitamin C. And I think you can definitely use IV vitamin C in conjunction with intratumoral. But I think the future of vitamin C, it is the intratumoral vitamin C delivery method. To use it in conjunction with intratumoral immunotherapeutics, like what we do at the Williams Cancer Institute. And we do intratumoral chemo and intratumoral immunotherapeutics, both immune checkpoint inhibitors and low-dose chemo. And by going intratumoral, we could actually lower the dose of the vitamin C. So it's going to reduce the adverse effects. You can have off-target toxicity with chemoimmune checkpoint inhibitors. Vitamin C has been shown to reduce that, but we can also put those together at lower doses, keep that in the tumor, and really limit, all off-target toxicity effects. Empower the immune system to do the job locally, train it, harness it locally, and allow it to spread systemically. That's the beauty of the apiscopal effect. And I believe intratumoral vitamin C is right there. It's the power of vitamin C, but that's the power of this new delivery methodology that I believe you're going to see coming to you very, very soon. So look for more information on this at the WilliamsCancerInstitute.com, because that's probably going to be where you find that information. If you want to hear more cutting edge information, more study deep dive on all things vitamin C and all things integrative oncology or integrative medicine in general, go over to pre-scribe.com. That's the Prescribed by Dr. Nathan Goodyear website, where you'll find the Prescribed podcast, the Prescribed by Dr. Nathan Goodyear podcast, where we do all deep dives here. You also find blog posts. You'll also find all of our videos over at the YouTube. So check us out there. I encourage you to please share this with people that would benefit from this information. And that includes researchers, that includes doctors, that includes patients. If we empower patients, that's how change happens. We need your help to share this, to grow the awareness of integrative therapies, because most conventional doctors don't know. They don't read because they're not asked to read outside of their current continuing medical education process and strategy that's created for them. And I also encourage you to drop us a 5-star review and share, like, and subscribe this if you find any of this beneficial, or on social media. Help us force the change of the future that empowers patients to receive therapies, not that destroy the body, but that heal it. That don't go to war and turn the body into a battlefield, but target the tumor and heal the body and never create the battlefield. That's the power of integrated medicine. That's the power of evidence-based integrative oncology. We'll see you on the next Prescribed by Dr. Nathan Goodyear podcast. Thanks for joining us on this episode of the Prescribed Podcast. All references supporting this episode can be found on the prescribe.com website. That's pre-scribe.com. To get your next dose of medical innovation, make sure you subscribe to Prescribe. We'll see you for your next refill.